Today I'm going to be telling you about some of the latest work that we have been doing on adenovirus assembly and structure. Now, adenoviruses are not the first thing that comes to mind when you think about viruses causing disease. Actually, adenoviruses are best known as therapeutic vectors. For example, you can see here, and you probably already know, that many of the most advanced vaccines against the SARS-2 coronavirus are based on the use of adenovirus vectors. However, adenoviruses are actually pathogens. As you can see here, they are the cause of a large proportion of diarrhea cases, even causing death in children. Adenoviruses can also cause economic losses because they also infect poultry, sheep, or cattle. There are cases reported of very serious pneumonia in individuals, immunocompetent individuals. And in cases of immunocompromised patients, the cases get even worse. Here, for example, a couple of years ago, there was an outbreak of adenovirus infections in a clinic in the United States, and 11 children died. And they are a serious threat in the case of patients undergoing transplantations, either solid organ transplantations or stem cell transplantation. As you can see here, there are many infections caused by adenoviruses in these patients, and the mortality rate is quite high. At present, there are no drugs to treat adenovirus infections that are specific and efficient, and the non-specific drugs that are used they do not work that well. So there is a certain need to develop treatments for adenovirus infections. And in this audience, I don't need to tell you that the structural data are key to develop efficient drugs. And there are many things that you can look at regarding structure and infections in the cell, but we are focusing on these two aspects. We are focusing on the virus assembly and the structural variability of adenoviruses. That means what makes some adenoviruses different from others, and why do they cause different diseases. So regarding adenovirus assembly, we've been working for many years, more than 10 years now, on adenovirus assembly using a combination of all sorts of electron microscopy techniques together with atomic force microscopy in collaboration with the group of Pedro de Pablo. But first, let me show you what an adenovirus, how an adenovirus looks like. Adenoviruses are double-stranded DNA viruses. They have a non-enveloped icosahedral capsid. As you can see here, this capsid is rather large and complex. It has a pseudo-T equals 25 triangulation number. The facets of the icosahedron are made mostly by the hexon, the major code protein. At the vertices, we have the penton-based protein and the fiber, and these are responsible for binding to the receptor. And then, in this case, this is human adenovirus type 5, the most commonly used vector. To assemble an Ad5 capsid, we need a series of minor code proteins. Some of them here are located on the inner capsid surface, and some others are located on the outer capsid surface. And here I want to highlight this protein, protein 6, located inside the capsid. This protein is interesting because it has membrane-lytic activity, meaning that during entry in the endosome, this protein needs to come out of the capsid so that it can interact with the endosomal membrane to allow adenovirus escape into the cytosol so that it can keep traveling all the way to the nucleus. Inside the icosahedral shell, we have what we call the adenovirus core. The core contains the double-stranded DNA genome and a large amount of virus-encoded proteins. These proteins have a large positive charge, and so it has long been assumed that the role here is to screen the negative charges of the DNA molecules so that the genome can be squeezed to be packaged inside the capsid. And here I want to highlight, because I'm going to tell you about this protein later on, the most abundant core protein, protein 7. And also another interesting thing in adenoviruses is that the maturation protease is also a DNA binding protein. It is packaged together with the genome, And it uses the DNA as a track to slide on, searching for all its targets on inside the adenovirus particle. And these targets include many of the core proteins and also some of the internal minor core proteins. So as I was saying before, we've been working on adenovirus assembly for more than 10 years now, and we've come up with this very schematic, very simple model for adenovirus assembly. First, we have the genome, and the genome is condensed with the help of the core proteins. Then also about the genome, we have L15255, which is a packaging protein. This protein is required for packaging. L15255 can bind both to core elements, to core components, and to capsid components. And it can also form homo-oligomers. So we think of L152 as a Velcro protein, helping to put together the core with the capsid during adenovirus assembly. So capsid fragments would be recruited onto the condensing core, and the capsid would grow, and eventually the maturation protease, which has been packaged together with the genome, starts cleaving its targets. And the first target it cleaves is the packaging protein, which is released and is not present in the mature particle. Some mutants get stalled here at this state of assembly, and they produce what we call immature particles. These particles are not infectious because they are much too stable. They do not properly uncoat, and that means that they do not release the lytic peptide, and they become trapped in the endosome. Now, when the protease cleaves all its targets in capsid and core proteins, we have the mature particle. The protease, among other targets, cleaves the core proteins. And we've seen that the cleavage of the core proteins somehow decreases their DNA condensing activity, so that now the repulsion between the DNA strands increases, and this creates an internal pressure in the particle. So all in all, this makes the particle metastable, primed for uncoating, so that when it receives the correct stimulus, such as binding to the receptor or acidification in the endosome, the weakest part of the capsid, which are the pentons, gives way and opens a hole through which the lytic peptide can be released for endosomal escape. And finally, at the nuclear pore, the particle is completely disassembled, and the genome, together with the core proteins, is inserted, enters the nucleus. So we had this model, and we were very happy with it and very proud of ourselves. But surprisingly, a few years ago, Patrick Hearing in Stony Brook University showed that you do not need the most abundant core protein to package the genome inside an adenovirus particle. So it seems that this DNA condensation activity of protein 7 is not required for packaging. However, what they observed is that these 7- particles are not infectious. And they also observed that the 7- particles in the cell become trapped, stay stuck at the endosome. So it seems that protein 7, core protein 7, would be playing a role during the virus entry. Now, to figure out what was happening here, we started a collaboration with Patrick Hearing and Urs Greber, and we looked in more depth at the behavior of the 7- particles. The first thing we did, or actually Marit Suomalainen did in Urs Greber's group, was to see if the lytic peptide is released in the cells. And the way to look at this is to label protein 6 with antibodies and the capsid with a fluorophore. And what Marit observed was that in the wild-type infection, when the virus enters the cell, the signal for protein 6 increases, meaning that this protein becomes accessible to the antibodies. However, in the case of the 7- particles, this increase does not happen. So in 7- particles, the lytic protein 7 is not exposed on the outside of the particle. Then we thought, okay, so why is the lytic peptide not released? Maybe the 7- particles are much too stable, as I told you before, for the immature adenovirus particle. So we looked at particle stability using extrinsic fluorescence, looking at what temperature do we need to use to make the genome exposed to the solvent. And we also looked at mechanical stability of the virus particles. This is something that we do in collaboration with Pedro de Pablo here in Madrid. And in these experiments, we take repeated images with the atomic force microscopy of the same viral particle. And in this way, we can simultaneously induce and observe the sequential disassembly of individual adenovirus particles. And what these experiments told us is that the 7- particles are not much too stable. On the contrary, they are unstable. They open up at lower temperatures to expose their genome. They release pentons much faster, and they crumble down much faster than the wild-type particle. So the particle seems to be uncoating correctly, losing pentons and all that. So why is not the lytic peptide released? So here's when we started looking at the structure. And before showing you our results, I need to show you what we know about the structure of protein 7. And what we know is very little. Only this little piece here, this N-terminal peptide cleaved by the maturation protease, has been traced in cryo-EM maps of human adenovirus type 5 obtained by the group of Hongshao. And you can see here this little peptide. Interestingly, Hongshao also found the N-terminal peptide of protein 7. This is the lytic protein, bound here. They are both bound inside this cavity in the hexon. Down here would be the virus genome. And the interesting thing here is that this is a trimer, a hexon trimer. Both the peptide of P6 and the peptide of P7 bind to the same site in the hexon monomer. So Hongshao proposed that we would be competing for binding to hexon. These two proteins would be competing for binding to hexon. Okay, so we obtained cryo-EM maps of wild type and 7- particles. One thing interesting here that I was forgetting is that this site here is the cleavage site, where the maturation protease needs to cut these two proteins. But as you can see here, this is the protease. This cleavage site is hidden, is recessed in this cavity. So there is no way that the active site of the protease can access its target site. Anyway, so we obtained cryo-EM maps of wild type and 7- particles and calculated difference maps to see if we could detect where we were lacking density, which should correspond to protein 7. And indeed, the difference maps showed this little red difference blob here that overlaps perfectly with the yellow strand where Hongshao had traced the N-terminal peptide of 7. If we now turn this around to look at the 4 hexon trimers in the asymmetric unit from inside, we again see this little density blob here, and this is where Hongshao traced protein 7. But you can see that there are similar blobs in the other hexons where there is nothing traced in the Hongshao structure. But these are located in the equivalent sites to this one in the hexon monomer. And if we keep lowering the threshold to render the difference map, we start seeing differences in all the equivalent binding sites. So this is telling us that protein 7 can bind to all the possible binding sites in the hexon in the asymmetric unit, confirming the hypothesis that protein 7 and 6 are competing for the same binding site in hexon during assembly. Now, when we looked at the sections of the maps, we observed that in the 7- particle here in the hexon cavity, there is some extra stuff here that is not present in the wild-type virus. This was surprising because we are looking at 7- particles. They are lacking 7, but they are not supposed to have any extra components there. The first clue we got about what this extra density can be came from the paper describing the mutant by Patrick Hearing, where they had observed that in the absence of protein 7, protein 6 is not completely cleaved by the maturation protease. They saw that out of the 2 cleavage sites in protein 6, the N-terminal cut is not made when protein 7 is not present. Now, when we look at this extra density here inside the hexon cavity, you can see that we have this tubular shape, and this extra density connects directly to the N-terminal peptide of 6 that Hongshao had traced. So it seems that we are indeed seeing the next part of the protein because there has not been a proteolytic cut here in these particles. Now, the interesting part here is that this next part of the sequence in the protein contains the amphipathic helix that has the membrane lytic activity. And indeed, we can fit them as some standard alpha helices. So we believe that we are seeing now the location of the lytic peptide inside these particles. And we came up with an organization, a model for the organization of protein 6 in the 7- particle. Here we would have a dimer. In this particular hexon, we have 2 copies of protein 6. And this would be the monomer putting together the pieces that Hongshao had traced together with our current observation. These data were obtained from cryo-EM maps at between 5 and 6 angstrom resolution. We have now a map at 3.2 angstrom resolution, and you can see that, yeah, there seems to be a helix there. However, this is weak density. This protein does not follow icosahedral symmetry. There is a variable occupancy in the different hexon trimers, so we are carrying out a lot of work here, doing local classification and reconstruction to see if we can actually trace a little bit more of protein 6. Nevertheless, what these results tell us is that when the core protein 7 is not present, the lytic peptide is trapped inside this hexon cavity. And because it is trapped here, the protease cannot reach its target site, and the lytic peptide is not exposed, and that's why these particles become trapped in the endosome. So this is our model for the role of core protein 7 during adenovirus assembly and maturation. We have the hexon with a binding site that can be occupied by either protein 6 or 7. You can see here by the copy numbers that we have more candidates to fill these binding site than binding sites in the capsid. So there is a competition between the 2 proteins. In some cases, protein 7 wins, occupies the binding site. So protein 6 is pushed out of the hexon cavity, and now it is accessible for the maturation protease to cleave it at its N-terminus, and also for the lytic peptide to be exposed and break open the endosomal membrane. When we do not have protein 7, all the sites are available for protein 6, and protein 6 is stuck here and cannot properly be cleaved and exposed during entry. So this is all done working on adenovirus type 5, but it turns out that there are over 200 different adenoviruses found in nature so far. They infect all different sorts of vertebrates, and we don't really have that much structural information about most of these adenoviruses. Even the human adenoviruses, there are more than 100 reported so far, and they can cause different diseases, and we don't really know exactly why they have different tropisms or different pathologies. So we decided to start working in some of these less explored adenoviruses, and we have recently solved the structure of human adenovirus species F, which is an enteric adenovirus. It causes gastroenteritis. And it is peculiar in the sense that this is its only tropism, unlike the other adenoviruses which have a main tropism but can also cause disease in other parts of the body. And we have also been looking at adenoviruses infecting lower vertebrates, and these belong to a different genus. These belong to the Adenovirus genus. This Adenovirus genus includes some important pathogens for sheep and cows. So let me show again the schematics of the adenovirus capsid organization, and now I'm going to focus on this protein, the external minor capsid protein 9, which has been traditionally related with maintaining capsid stability. And you can see that in human adenovirus type 5, it makes this sort of network weaving around the hexons in the facet. So this is the structure of protein 9 in human adenovirus type 5. It has an N-terminal domain that we call the triskelion domain because in the capsid, it makes trimers with this triskelion shape. Then we have the rope domain. The rope domain is flexible and mostly it cannot be traced in the structures. It connects the triskelion with the carboxy terminal alpha helix. And in adenovirus type 5, these helices form this 4 helical bundle here at the capsid edges with 3 helices coming from copies of 9 from the same facet, and the 4th helix comes in the opposite direction coming from the adjacent facet. This is human adenovirus. It turns out that in non-human adenoviruses, the rope domain is much shorter. And the alpha helix is directly next to the triskelion domain. And so now we have a 3 helix cluster located directly above the triskelion. So there are different conformations in human and non-human mast adenoviruses for protein 9. We have now solved the structure of enteric adenovirus, human adenovirus type 41, in parallel with a group in Sweden. And you can see here that we have the triskelions of protein 9. There is one at the icosahedral trifol axis and this other at the local trifol axis here. Now, in our structure, we can trace the triskelion domain, but we see no density that we can use for tracing at high resolution for the rope domain or the carboxy terminal alpha helix. However, we do see weak blurry density that could correspond to this part of the protein which is not icosahedrally ordered. And this weak density shows that there is a connection between the central triskelion and the local trifol axis, the triskelions at the local trifol axis. And there is also blurry density located directly above the local trifol axis triskelions. So we interpret this weak density like this. We think that the rope domain of the central triskelion extends away from the center of the facet to join the C-terminal helices in the local trifold triskelions here at the periphery of the facet. And they might form a helical cluster here, but this cluster must be mobile or unstable so that we do not see order density for this cluster. So it seems that protein 9 in the enteric adenoviruses is different, has an organization different from all those previously observed in either human or non-human adenoviruses. And this also is telling us that the rope domain, which is slightly shorter here and much shorter here, and also has differences in sequence, is highly variable. This rope domain dictates the organization of this external decorating protein in the different adenoviruses, because it can turn clockwise, like you see here, or counterclockwise, or here it seems to go straight to encounter the triskelion here at the periphery of the facet. Now, it turns out that protein 9 is only present in the mast adenovirus genus. In other genera, there is no protein 9, but the adenoviruses have a structural protein that kind of substitutes for protein 9, and this is called LH3. Now we have solved the structure of an adenovirus infecting lizards, and you can see that LH3 forms these purple blobs here on the very outer capsid surface on top of the hexon towers. Several years ago, in collaboration with Mark van Rijk, we showed that this LH3 protein folds like a trimeric beta helix. This fold is very typical of tail spikes, receptor binding proteins in tail phages. In 2017, we only had the structure for this domain. It was recombinant protein. Now we have the protein in the context of the capsid, and we can see how the beta helix domain connects with the capsid floor by this domain here. And let me remind you that LH3 is substituting for 9, and it is occupying the same sites in the capsid where the 9 triskelion is found in the human and other mammalian adenoviruses. So let's compare LH3 with 9. Here I'm overlapping LH3. There is a hexon back there for reference. And in brown, we have protein 9 in the human adenovirus type 5. And you can see that the structures are very different. The AD5 protein goes all the way here to the capsid edges to form the helix cluster, while we have here on top of the triskelion this beta helix domain. But the part of the proteins that connect with the capsid surface is exactly the same, as you can see here. If we compare LH3 with protein 9, we have the triskelion shape here with this hydrophobic core. And if we look at it from the capsid surface, this similarity is seen much better here. So this similarity had not previously been found just by looking at the sequences, and it has now been revealed by the structural studies. And this means that this triskelion motif is conserved across genera and is important to bind the correcting proteins to the adenovirus capsid. And finally, let's look at these two proteins in the context of the virus genomes. We have here in the mastadenoviruses protein 9 next to a protein called E1b55K, which is not a structural protein. This protein is a multifunctional protein which is expressed early in infection, and what it does is mainly it deregulates the cell cycle. In the adenoviruses, we have LH3, but when this protein was first discovered, it was called E1b55K also, because the sequence similarity between this and this is very high. Now, what we see is that in the humans, we have E1b55K, and downstream, we have 9. In the reptilian adenoviruses, we have this part of LH3, which is very similar in sequence to E1b55K, but it has a triskelion here, which is very similar to the triskelion of 9. So what's going on? Are bits and pieces of the genes jumping around in the adenovirus genomes, or what? So to answer this question, we contacted Mart Krupovich in Paris, and Mart did an exhaustive analysis of and comparison of all the adenovirus genomes, and he found that the E1b55 or LH3 genes are present only in these two genera, adenovirus and mastadenoviruses. But it turns out that at some point during evolution of adenoviruses, the LH3 gene was duplicated, and actually there are some mammalian adenoviruses where and these are found in horses, that still keep the 2 copies of the LH3 gene, one that has already lost the triskelion and the other one still keeps the triskelion. In the human adenoviruses, the copy that lost the triskelion acquired this long N-terminal extension and became the oncoprotein E1b55K. The other copy lost all the rest and kept only the triskelion and became protein 9. So in conclusion, what I wanted to tell you today is that there is a certain need to develop anti-adenovirus drugs. Core proteins are not just passive stuffers helping to squeeze the genome inside the capsid. They are playing active roles during maturation. And then adenoviruses have triskelion-containing decorating proteins that are markers of adenovirus evolution, and may also have a say in host specificity, since they are there on the outer capsid surface, presumably interacting with host factors. And they are different in adenoviruses with different tropism. And that's it. These are all the people involved in the different projects with color coding. And thank you all for your attention.
Thank you very much, Carmen, for this very nice presentation.